I just started recording. Well, good morning, everybody. Welcome to this lecture. I am basically talking about sequencing technologies today, and I'll get started on the coding part. Basically, the three of your projects is going to rely a lot on the things that we'll discuss from this week on. And again, I want to reinforce the idea that you guys are giving a presentation. I believe it's on week 8, right? I think it should be before August 10. Do you know the exact date? Is it August 14 or something like that?
I think it's August 14th.
Yes, yeah. August 14. I said before August 10, but I had August 15 in mind. Uh, okay, so your final presentation is August 14. Today is the 15. We have a month left. We have basically 4 weeks. In my mind, it's more than enough for me to give you the full picture of your projects as far as the coding in the command line goes. And Lucia she told us that she has experience dealing with Python and GitHub. And this can be really, really challenging, can be really, really complex. So you'll see, we'll discuss everything. But now I will just get started on the DNA sequencing technologies. Just share my screen. You all see my screen, correct? All right. So I chose the title here, DNA sequencing evolution, because I wanted to give you an overview of 60 or 70 years of development, and I only have probably half an hour to do that. So let's get started. So the plan for this is that I'll have basically 3 parts. It's going to be a brief discussion on the 3 big DNA sequencing technology groups: the Sanger from the 70s to the 2000s, sequencing by synthesis, Illumina, starting around 2005. These dates are approximate, they're not exact, so people could argue that this is wrong, but I'm just giving you an idea. And then PacBio and Nanopore starting in 2015 and beyond. So in the second part, I'm going to revise PCR and primer design because these are basic steps in DNA sequencing. And we'll have a primer design practice where I will show you how to do an in silico PCR using the UCSC genome browser. Finally, in the end, I'll show you some documents that I have prepared describing each of your projects, BME01, BME04, and BME03. Sorry, BME05. 3 here is a typo, individually. I don't know if you all noticed, but I included the documentations in mine, Jaime's, and Danilo's folders. And that documentation is going to be regarding the bioinformatics / coding aspect of your project. And that's my area. At this point, it's my area of expertise, and I will be mentoring you, the three of your projects. I am the secondary mentor in all of your projects. I hope you realize that we have this Recombio tutorials. The idea is to offer this Recombio stands for Research in Computational Biology. Frankly, I really need your help on this because we decided that this would not be offered only to you with me here, my regular SIP audience. This would be shared with a broader a wider SIP audience. So I was not expecting that there were 60 people registered for the English version of this. and I have a particular interest in a Spanish version of the research in computational biology tutorials. But there are not many, at this point, there are not many Spanish speaking people interested, but I believe that's going to change. And as I was discussing earlier, it looks like I will do this through GitHub because there are facilities associated with the things that I have in mind. And I will let you know, and this will probably increase our collaboration capacity, and we'll learn, frankly, we will learn together how to deal with this. So I'll just show you today the R Markdown notebooks. I will not go into coding today because the art tutorial sessions will be held next Tuesday, the 20th, Thursday, the 22nd, and the Tuesday after, the 22nd, yeah, and then the 27th, which is the Tuesday after. The thing that I need to say up front is that although I would love to, I do not have the means to help people that much figure out their computational infrastructure that they need in their own computers. So I'll just give you an overview, and then you'll have to do lots of search and lots of research, and I'll try to help as much as possible. So I need to expect that you have at least R and RStudio installed. And I asked last week, majority of you have a MacBook Pro, a MacBook, and I am expecting, it doesn't mean that it will happen, that you can have access to Bash using R Markdown. Any questions about the plan, or can we just get started on the sequencing part? Any comments, anything?
I just wanted to ask, the art tutorials, will they be like during our usual meetings, or will they be in the afternoon?
Yeah, the thing that I had in mind is that you guys are paying us for this time. So at the very least, we'll give you the regular hours. And if you need more, I can offer more. But right now, what I have in mind, what I have planned is on Tuesdays and Thursdays, offer you the coding in the regular time here, and let's coordinate if you guys need more help with that, because I think we should have started this earlier. What I'll do is I will hold a session tomorrow at 10, and I know that there is another SIP activity that conflicts with that you might attend. I was actually asked to encourage you to attend that other I think it's the research, writing a paper workshop, right? Does anyone know about that? What is it, Kira? Can you remind me?
I have like my calendar right here, and it's writing effective papers. Yeah.
Yeah, it's at 10 a.m. tomorrow.
Mm-hmm. Yeah.
Okay. Yeah, so I was actually planning on attending that and helping Raja, but I'll have that conflict, and maybe we can arrange the time. But as of right now, because I am telling you, maybe we should discuss to change that that time. But the problem with that is that I already sent a message to people that are in Mexico, so there is a chance that they are expecting that to happen. So I don't think I can change that. But definitely for Monday, I'll have another session on Monday at 10 a.m. California time. It's going to be 12 p.m. at my time here in Chicago. And I hope that that will help people that need to have their computational structure figured out. Kira, go ahead and ask.
Yeah, I had that question about like the Friday session versus the Monday session. Are they both the same, but just on different days?
Yeah, because I basically want to see where you guys are in terms of your computational structure, to see if you have the minimal requirements for Tuesday.
All right, so would you encourage us to go to the workshop on Friday and then come to the software thing on Monday?
Sure, if you want to do that. What I told Sierra is that if for instance, nobody joins tomorrow, I will not be able to help anyone because I'll not have questions to deal with. I will not have people telling me, oh, I use a Windows machine. Although I use a Mac, I cannot use Bash. So I'll not be able to help with that, or I'll not be able to know that that is a problem.
Okay. I also had another question about the sessions on the 20th, the 22nd, and the 27th. It looks like on the SIP calendar, it said it was going to be at 3 o'clock California time for the English version on Tuesday, but would that then, so what are we doing
That's
at
not you.
at
That's not
10?
you. 3 p.m. is not you.
Oh, that's for other people?
Yes. You
Okay,
guys
so
are,
we have it at 10.
yes.
Okay, cool.
So that there's going to be 3 sessions on Tuesday, Thursday, and the next Tuesday, the Tuesday after.
All right, thanks.
Yeah. Hi, Jaime, welcome.
Hello, good morning.
Good morning. Okay, nobody has other questions or comments on here? All right, let's move forward. So why do we need to know about DNA sequencing? It is because a huge amount of information is present in the genetic material of any organism. I think we discussed that SARS-CoV-2 has a genome of 3K base pairs. As humans, we have a genome of 3.3 base pairs. That means 3.3 gigabytes of data. So our genome is roughly 100,000 times bigger than the SARS-CoV-2 genome. So both the virus and our genetic code contain the recipe for the construction of an organism. So what you guys are, what I am, in terms of the proteins, for instance, that make up our bodies, that information is encoded in our genetic material, in our genetic code, and that has been passed to us as the result of a long period of adaptation of our species in the earth that we live. So DNA sequencing is the starting point of any type of DNA sequence analysis. And last week, Danilo showed you how to build the phylogenetic analysis using Mega, I believe is the name of the software. So you got introduced to the concept of a FASTQ file and a FASTA file. These are very important sequencing file formats that we'll discuss a lot, I believe, in the weeks that are ahead of us. And we will expand on the notion of the files that we analyze. We're going to talk about VCF files, SAM files, BAM files, things like PAD files, and genome browsers that are tools used to visualize genomic information, and other tools that we'll cover. So I told you that I am relying on this paper for the description of these sequencing technologies. So this is a very brief, high level. It's like a timeline of the development of the sequencing technologies. Basically, here they bring to us that the insulin sequence was established by Fred Sanger in the 50s. In the 50s, and then the same Fred Sanger developed what's called the Sanger sequencing in the 70s. In the 90s, we have the advent of this spiral sequencing technology, which is considered a next generation sequencing technology. And I have to say here that one of the inventors of the pyrosequence technology was my PhD advisor, Dr. Nader Porman, who is also a professor at UCSC. And then after that, we have the begin of the technology that is the main or the major technology available for sequencing today. This is also called next generation sequencing. A more technical way to refer to this sequencing is sequencing by synthesis. And this relies a lot on the mechanics of PCR that we'll discuss today. Here, basically, I'm talking about the Illumina sequencing technology. And the Illumina sequencing technology probably is used by 90% of the sequencing market. And then they talk about detection of DNA methylation. And the reason why I highlighted this here is because I now work with epigenetics. So it's modifications, not, I want to ask you this question. What do you guys understand about epigenetics? What is epigenetics? What is genetics and what is epigenetics? Go ahead, Kira.
Genetics is about the code itself, while epigenetics is about the expression of the code. So I guess different factors can lead to the transcription of some parts and the restriction of the transcription, like through the, I guess, how easy, like whether the bind, I guess is the, I think my understanding is that whether the histones are bound tightly or not affects the amount of ease with which the gene can be transcripted and translated.
Thank you. Very good explanation. Lucy?
Yeah, so I sort of agree with Kira, but like in a more general sense, epigenetics is sort of like how environmental and behavioral factors, they affect the way that like our body interprets our genes. And if I'm not wrong, most of the changes are not permanent, they may be reversible, but it's like that study of interaction.
Okay, thank you. Very good response too. I'm not 100% sure if the epigenetic changes are always not permanent. I'm inclined to think that some epigenetic changes are permanent. You guys mentioned expression, and when I hear that word, I think about RNA expression or protein expression. So RNA expression is not exactly on the, there's a field already for RNA expression, it's called transcriptomics. So epigenetics, I like, Kira, when you mentioned histone modifications. So epigenetics is really something that is on the side of the DNA sequence. It's not a modification on the sequence of nucleotides, but it is a chemical modification in the DNA. So here they bring in 2010, the detection of DNA methylation using single molecule sequencing. I'm inclined to think that they are talking about Nanopore or PacBio. And one of the great advantages of Nanopore compared to Illumina sequencing is that, for instance, Illumina sequencing does not, the sequencing process itself does not allow you to know if the base that is being added on each sequencing cycle, if that base is epigenetically modified or not. Whereas these more recent technologies such as Nanopore, I think we'll discuss a little bit more on how Nanopore allows identification of DNA modifications such as methylation. Okay. All right, so let's discuss a little bit of the mechanics of the first generation sequencing. This is a high level review of Sanger sequencing. Basically, this process, in my mind, you can understand this from the perspective of second generation sequencing. So this is a very general process. So in all of them, in the three big groups of sequencing technologies, you always start with genomic DNA or whatever genetic material you want to sequence. You fragment the DNA. In Sanger sequencing, there was this part here where people had to do cloning. So what you do here in this step is that you basically transform bacteria with a plasmid. Are you guys familiar with that idea, with that concept? Transform a bacteria. Transforms mean that you figure out a way to insert the plasmid into the cytoplasm of the bacteria. And that way is such that you do not kill the bacteria. It is allowed to grow, and it's allowed to replicate and generate what? What are you interested here? As the bacteria is replicating, what do you think is replicating with the bacteria that you might be interested in?
The fragmented DNA.
Exactly. Here the idea is that you're amplifying the plasmid that was introduced to the bacteria, and you're generating huge quantities of the genetic material. And this type of technology relies on the amount of DNA that is being analyzed. So here, my understanding is that you need huge amounts of DNA compared to what we do today, where we use way less. My understanding, way less DNA than in these days. So after that, basically we have the sequencing, which relies on DNA synthesis. There's another name for this, and I just forgot. DNA synthesis, the addition of nucleotides. So every time a nucleotide is added, in the case of Sanger sequencing, you're going to have a competition between a normal nucleotide and a dideoxynucleotide. The dideoxynucleotide does not have hydroxyl group that allows the chain to grow. So once the dideoxynucleotide is introduced, the reaction stops, and that fragment cannot be further extended. So in the detection step, what you do is you basically detect, in this case, I believe it's fluorescence that these nucleotides are labeled with. So that fluorescence can be detected in the instrument. Any questions, any comments here? Jaime, please feel free to add your thoughts if you think it's necessary, okay?
Okay, Joe, you can go ahead. It's fine.
All right, this is a more deep, how can I say? This is a more thorough overview of what Sanger sequencing technology looks like. So as I said, no, I didn't say this, but Sanger sequencing is also known as chain termination sequencing. So it is based on the addition of a chain termination dideoxynucleotide by the DNA polymerase during DNA replication. I was trying to remember the word replication rather than DNA synthesis. So after the sequence, after the replication or the synthesis, the fragments are resolved in gel electrophoresis, and that's shown here in the right, the electrophoresis, and the detection of the nucleotides is done by using fluorescence, as I mentioned before. So what's going on here is that you have the DNA synthesis happening, and DNA polymerase is adding these nucleotides all the time. Both of the normal nucleotide and the dideoxynucleotide are fluorescently labeled, and you see, okay, what happens if the nucleotide that's being added is not it's small here, but you can see that the normal nucleotide has a hydroxyl group. The dideoxynucleotide does not have a hydroxyl group. So what happens when the DNA polymerase adds a normal nucleotide? What happens in terms of the ability of the template to grow? What's going on? And Brit, is that how you say your name, or is it just Anne?
Oh, yeah, Anna Britt.
Anna Brit. Do they call you Anna?
I don't know. Usually people just call me Anna Britt.
Go ahead, Anna
Yeah.
Brit.
For the normal nucleotide, at least, the DNA sequence will keep on replicating. But when you have the one without the hydroxide group, then that will, then the complementary chain, or like the DNA sequence will then terminate
Yes.
because it can't, yeah, the nucleotide can't continue on.
Perfect, yes. So what happens is that you have the generation of fragments that have a distribution of sizes, and these sizes are due to the fact that some of them received the nucleotides with the hydroxyl, and therefore they could grow. And some of them received the dideoxynucleotide, and therefore they could not grow, and the sequencing is terminated, it ends. So if this one received the dideoxy, it ends like this. And it turns out that this one is read in the gel electrophoresis in a region like this. And I am assuming the migration is happening from bottom to top, as far as the arrow indicates. So one short fragment like this would end up here in the north, as opposed to the bigger fragments that ends up here at the south. Does this make sense? So, well, when I was learning about this, I used it to hear people saying that in the beginning, they did Sanger sequencing by using huge gel electrophoresis. My impression is that this changed over time. Later, they used capillary gel electrophoresis, but this sounds to me like something that was a development of big gel electrophoresis. So I'm just trying to say that of course, there was development in the technology, and I'm also trying to tell you that I'm not as old as someone that has worked with these big gel electrophoresis in the beginning, I believe, of Sanger sequencing development. Okay, I guess we covered Sanger sequencing. Now let's discuss second generation sequencing. And I'm going to tell you up front that a difference between Sanger and second generation is they refer to this in this paper because they refer to second generation as massively parallel. Another way of referring to this is high throughput sequencing. And I want to ask you, what is then the difference between this second generation and the first generation? What do you think might be the difference? Thoughts? And Brit?
This is a bit of a guess, but is it the adapter ligation that's different?
Yeah, I don't think they use adapters in Sanger. They use primers, which have a similar role. But that's not correct, actually. But a good guess.
Okay.
Good guess. Lucia?
So what I understood from the paper is that the next generation sequencing is much more effective. Like it can sequence at a much faster rate than the Sanger one.
Yeah, so that's the point. Kira? Sorry, I hadn't seen Kira.
Oh, I was wondering about the name massively parallel, whether it comes from what's going on in the amplification process, which is slightly different from the original, the first generation idea.
I'm trying to see if I understood what you said. Yeah, I agree. I agree. Here you have cluster generation. So here, I think the term here is the amount of data. And here is, we need to introduce this concept of big data. Did you ever hear this concept of big data? Huge amounts of data. You're gonna see people talk about the number of reads that are generated. Sorry, that are generated in each sequencing run. So for instance, the Illumina MiSeq generates an average number of reads around 25 million reads. So what 25 million reads means is that there are 25 approximately, right? These, I am talking about approximate numbers. There are approximately 25 million spots here, 25 million spots, each with something like this, Kira, when you talk about bridge amplification, we are talking about a process that, did you talk about, you talked about cluster generation, right? Did you mention, I can't remember if you mentioned cluster generation, but cluster generation is a process that happens using bridge amplification. So what's happening here is that you have the oligonucleotides on the surface of the flow cell. Do you remember that I mentioned that this is what a flow cell is? It's basically a glass slide with oligos on its surface. And what you do is that you prepare and you bind the adapters to your DNA of interest. And these adapters are complementary to the oligonucleotides. So once you have your DNA of interest bound to the flow cell, to the machine, you have the process of bridge amplification and cluster generation. So why do you think we have to have a cluster generation at this process here? So this is a cluster, this is another cluster, another cluster, another cluster, another cluster, another cluster. So in cycle 1, you have the detection of a cytosine. In cycle 2, thymine. Cycle 3, guanine. Cycle 4, a adenine. So what is this? This is a read that is being generated. The average size of this fragment here for an Illumina machine is less than 1,000 base pairs. So in Sanger, I believe the size of the read that is generated is also less than 1,000 base pairs. The difference in this process is that we are reading 25 millions rather than one sequence. Does that make sense? So in each cluster, we are reading one sequence. That's why it's called massively parallel or high throughput sequencing. And the word parallel here really means parallel, because this one cluster is parallel to this, which 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 means that in the flow cell, they are side by side. They are parallelly distributed, if that makes any sense. Okay, questions or comments here? So again, the difference between first and second is the amount of data that is produced. And now we come to the third generation. And I cannot tell you a lot about this because I have never worked with nanopore sequencing, for instance. But the technologies that we discussed relied a lot on PCR. And that's also the reason why I am doing the primer design and the PCR review today, because basically what I'm trying to say here is that these technologies relied a lot on the interaction between DNA polymerase and the DNA template, which means the amplification of DNA. The problem with that is that these enzymes have errors. So there are errors associated with the identity of the nucleotide that's being incorporated. And there are also biases that are related to the sequence, the region that they are sequencing, which means the biases are related to the nature of the sequence. And so, and there is information loss. For instance, if you're sequencing something that is near the 5' or the 3' region of your fragment of interest. And oh, sorry, here they're talking about the methylation. As I said, Illumina sequencing does not allow you to record information about methylation because it doesn't have the means, as far as I understand, it doesn't have the means to record that information. And the way that I want to talk about the means, when I say the means, is I want to introduce this idea that is used in Nanopore and PacBio. For the sake of simplicity, I'm not going to talk about PacBio sequencing. So the thing about these new technologies or these newer technologies is that they sequence each molecule at a time, or in each pore. Here, I learned this with Jaime. In each pore, I don't know if this is still called the flow cell, but in each pore, what's happening is the translocation of a single-stranded DNA. And what also happens is that as the DNA trespasses the pore, there is a change in the electric current that is characteristic of the identity of each nucleotide. Now, the way that this type of technology allows us to read modification in the nucleotide identity is because a cytosine, for instance, if this cytosine is methylated, the change in the signal is going to be 1. If the cytosine is not methylated, the change in the signal is going to be different. And therefore, it is possible here, what is possible here is to record what the sequencing signal is from a base that is methylated and a base that is not methylated. So that's one of the differences and advantages of Nanopore, of this third generation compared to the second generation. The other advantage is the size of the molecule. So when we talked about Illumina sequencing, we were talking about around 1,000 base pairs long reads. Here we are talking about 20,000, 30,000. I am not entirely sure if we are talking about 100,000 base pair long reads, but these technologies are also called long reads because they have the ability to produce long sequences, or the algorithms that are used to deconvolute the signal that comes from the trespassing of the base pairs through the pore. The deconvolution of those signals is going to be transformed into a fast A sequence, for instance, and that can store methylation data, for instance, or DNA modification, for instance. What's not recorded in Illumina sequencing, for instance. Questions, comments, or thoughts on this? Kira.
I had a question about the previous generations a bit. So I remember from the paper that I think between the first and the second generations, there was something called shotgun sequencing, maybe. So where exactly does that fit, and how did it go from shotgun sequencing to second generation?
Well, nowadays, what I call shotgun sequencing is next generation sequencing, and I'm not entirely sure how shotgun sequencing was done prior to Illumina, for instance, because my understanding of shotgun sequencing is what they call small read sequencing. Basically, you have a huge number of small reads that cover a big region. But on
Oh.
general,
Wait, so is it closer to second generation sequencing then?
yes, my understanding is that.
Okay, yeah, cool.
Mhm. Okay, now I want to talk about this other figure. This is really computational. This is computational, which is genome assembly. What is genome assembly? And since the question is already there, what is de novo? What does that mean? Did anyone raise their hand? Oh, sorry. Sometimes you need to talk to me because I'm not looking at the. I can't see who has lifted their hands. Kira.
It means like of new or like completely new. So maybe it's like starting from scratch.
Yes, yes, that's what I was going to say. Go ahead, Lucia. Sorry.
No, I was going to say the same thing.
Yeah, perfect. The literal translation for the novel in English is from scratch. In the Latin languages, that means again. But the literal translation is from scratch. So de novo genome assembly means that you are looking at a genome that has not been sequenced before. You isolate the DNA of that species, either from the blood or tissue or a cell. You isolate that DNA, you sequence that DNA, and here we are going to use NGS in general, right? We're probably going to use Illumina. So we're going to use this short read sequencing to assemble the genome. So we are going to look at the parts of the sequence that overlap, and we're going to build these contigs. So here we have 3 sequences that are overlapping, and we generate a contig, which means a bigger sequence that is a continuous sequence of the previous overlapping sequences. What happens is that sometimes these contigs do not have a continuous connection, and that forms what they're calling here this long-range links, and that generates the concept of a scaffold. So the scaffold is the connection of these contigs that are generated from assembling the short reads. And that's how we get to have information about the chromosomes in human, right? But assembly is also done for viruses, for bacteria, for plants, and for mammals. Any questions, any comments? Okay, so we need now to contrast de novo genome assembly with resequencing. So what is it meant by resequencing? Why is it called like this? Why is it called resequencing? One was called de novo, the other one is called resequencing. No guesses? Oh, Kira?
Oh, it's like in contrast to de novo, resequencing would refer to something that's already been sequenced before.
Correct, yeah. So the assembly of the human genome is something that was my understanding is difficult. There were several groups working on that, and that was finished around 21 and 24, 2001 and 2004. So what we do now is resequencing, and here they are presenting to us the areas that benefit from resequencing. So almost 6 years ago, when we had my son, we actually did this non-invasive prenatal testing, and we got to know his gender by sequencing. So the most, they say here, unexpected area of impact of DNA sequencing is non-invasive prenatal testing that is used in the United States, at least, for what they call family planning here in the United States, right? When to test whether a baby can be born with some genetic deficiency and things like that. And I guess we need to say this is used for pro-abort or non-abort policies. And I am not, that's one area that is that uses DNA resequencing. As I said, we also got to know the gender of our son by this method. And currently, my current work aims to find prognostic value in neuroblastoma resequencing using liquid biopsy from the blood of children and also from the tumors. So I'm using these models, I'm building these models using tumor RNA sequencing and epigenetic sequencing. So basically here I'm trying to tell you what the possible applications of resequencing, what they are. As you got from the paper, one problem, and for me that's a serious problem, is that currently this is mainly used for non-invasive prenatal testing and also for getting to know the gender of the babies and things like that. But I really have great expectations for the applications in disease. So now they bring any questions about the previous slide? Sorry. Comments? All right. Now they bring to us another table, a box, they call it a box, that is very, very interesting. It brings about the genome sequencing milestones and the computational milestones and the application milestones. So in the 70s, the first genomes were sequenced. So the bacteriophage genome was sequenced in the 70s. In the 90s, Saccharomyces cerevisiae. And then in 2001, Homo sapiens. If you didn't hear me say that, this had a huge contribution from UC Santa Cruz. From I guess I can say that it is the Department of Biomolecular Engineering and Bioinformatics. Professor David Hossler led that effort. And then in 2004, Rattus norvegicus, and I am highlighting that here because I worked with Rattus norvegicus RNA sequencing. And then in 2010, the Neanderthal genomes were sequenced, and these hominid sequences started to be, that allowed the study of ancient DNA from these hominid species. Interestingly, in 2013, the HeLa cell line was sequenced. I don't know if you know, HeLa stands for Henrietta Lacks, which is an African-American woman whose cancer, I think it was cervical cancer. She had her tumor cells isolated, and these cells have helped a lot in several areas of research. I can probably not name many of them, but things like vaccine, cancer, things like that. These cells were used. There's actually a movie on Henrietta Lack's life. That's why this is very important. Okay, genome sequencing milestones. Computational milestones, this is interesting for us because we're going to deal a lot with this. So in the 90s, the BLAST, the basic local alignment sequencing tool, which is a tool that allows alignment between sequences that Professor Danilo started to discuss last week. And C, from the 90s to the 2000s was when we had a company called Celera Assembler to, actually, I don't know if this is probably the name of the tool. I think the company is called Celera. This was a competitor of UCSC. Celera, as far as I know, is a company. UCSC is a public university. So what they did, they developed these tools for assembling the genome, and they were the competitors for the assembly of the human genome. And then in 2001, Bioconductor is I would say it's a suite of softwares. I hope I'll have a chance to show you how to use this in R. And then in 2002, the UCSC Genome Browser was developed. It's you can use it. I hope we'll have a chance to use the UCSC Genome Browser. And Ensemble Genome Browser, I use Ensemble a lot too. I think this is the Ensemble is from Europe, and UCSC is obviously from the United States. The NCBI Short Read Archive. Last year, we have a chance to use this for downloading COVID sequences. There are lots and lots and lots of sequences from human, from mice, from rats, probably from C. elegans. I have never used it, C. elegans. There are sequences from plants here, deposited here, that we could study. I'm not entirely sure if there are microarray sequences deposited here. That was developed around 2007. Then the Burroughs Wheel Alignment tool, BWA, we're going to use this tool. We are going to use SAM tools for detection of variants. We're not going to use Top Hat, but it's a tool that is used also for alignment. We will not use SOAP de novo, but it is it's a genome assembly tool. I don't think we will use the GATK, the Genome Analysis Toolkit. It's a suite of software. This one I'm sure, I know that it's called a suite of softwares developed by the Broad Institute of Harvard and MIT to allow, I have used it for identification of SNPs using sequences from the SRA, from the Short Read Archive. And then the integrated genomics viewer, I think I'll mention this again when I show to you the deliverables from each of your groups. Okay, now we turn to the application milestones. So the first genome sequencing was done in 77. That was the bacteriophage. And then in the 90s, in the end of the 90s, we already had, honestly, this is very new to me because I obviously do not know everything that happens in this field. At the end of the 90s, we already had the first human SNP arrays being studied, which means that at this point in time, people were already interested in the mutations that are developed from computational and sequencing analysis. In 2004, we had metagenome assembly. My understanding is that this is a genome assembly of microbiota, which means microorganisms that live in our gut, for instance, and inside the human body is my understanding. And then in 2007, we had the first studies that used chromatin immunoprecipitation. So that is the sequencing of epigenetic markers. I do not know if this was done with histone pull down or DNA methylation, for instance, but that's what this is telling us. Then RNA-seq is pretty new. Actually, epigenetics is pretty new if you compare with other advances, because RNA-seq is from 2008. And then 2007, the completion of the phase 1,000 genomes project. My understanding from this project is that it allowed the identification/study of many human genome sequences. And then we started to understand at this point how different populations, such as humans in Africa, humans in Asia, humans in Europe, humans in Australia, and humans in America, how they differ from each other, how these groups differ from each other in terms of the single nucleotide polymorphisms that they have or that they do not have. And then in 2001, I think it's a consequence of the 1,000 Genomes Project, we started understanding more about how these mutations are inherited together or not in the construction of these haplotypes or haplomaps, which are maps of SNPs, maps of mutations. I hope I'll have a chance to talk more about this to you. And then only in 2016 and 2017, we have the first studies using genome assembly using PacBio and Nanopore, which are the third generation sequencing methods. Comments? This table is really useful in my opinion. All right, let's now move on to primer design. That's a basic. Did someone want to say something? Okay, primer design. Primer design is used in PCR amplification. It requires a DNA template, primers, enzyme, and other reagents. I think I'll show you more. So the template is your analyte, the thing that you want to know about. So what you do, you hybridize the primers, and we'll cover a little bit of the molecular biology that goes after this. These are the considerations that you need to have in mind for primer design. I hope you guys saw this on the video that I sent. The primer length, the annealing and melting temperatures, the GC content of the primers, and also possible secondary structures on these primers. So here, basically, I'm talking about things that can be problems when you're doing an actual PCR. And if you guys had joined, if I had mentored you before last year, before 2020, when we had to all go remote, I would have a chance to show you what a PCR setup looks like. But if you have not had yet, I hope you'll have a chance to see what an actual PCR looks like. So the primer length, the optimal range is around 20 base pairs long. Less than that can give you non-specific regions, which means that the primers are binding to regions that it should not. But too long may give you problems in hybridization, because it's going to be so long that it's very hard to hybridize it, and it takes too much of energy for that to happen. So annealing and melting temperatures are really important, because this is basically the process of opening and hybridizing that allows amplification of your DNA of interest. So annealing allows base pairing, which is the hybridization. There's a typo here. Melting is the temperature at which half of the primers are dissociated from DNA. And a very critical parameter is that annealing should be 5 degrees Celsius below the melting temperature, so that the annealing can happen. If it's too high, it may not happen. And the two primers, the forward and the reverse primers, need to have similar annealing temperatures for the PCR to occur. The GC content has to do with the energy that is required for hybridization and denaturation, which are important in PCR extension. Secondary structures, these hairpins and heterodimers and self-dimers, they interfere. Well, heterodimers interfere with specificity or unspecificity of your reaction, and hairpins interfere with the RNA polymerase extension and the ability of RNA polymerase to move forward using the DNA template. So for our primer design, I want you guys to think about the concept of an amplicon size, and I want to ask you, what is an amplicon? Any guesses on what an amplicon is?
It's either like the region of DNA that's it's either the source or like what's the final product of the amplification process.
Thank you. Yeah, it's someone else. I cannot see if there's anyone else with their hands raised. Thank you. Yeah, the amplicon is the region that is being amplified. So the amplicon size, I'm trying to tell you about the length of the region that I want to amplify. I think we'll talk about regular versus quantitative PCR. No, I don't plan to talk about that actually. It's going to be too much time. So we are going to go now to an in silico PCR, and that's going to be using the UCSC genome browser. So the first thing that we're going to do, we are going to design our primers, and then we're going to do the in silico PCR using the genome browser, the UCSC genome browser too. Okay. What I'll do now, I will go. You all downloaded SnapGene Viewer, correct? All right. So my expectation is that you have done so. So I'm gonna go to here, SnapGene Viewer. I have my sequence, and as you can see in my map, I have several features here that I use. For instance, I use the sequence of the spike protein. I have the sequence of the NSP protein, the nucleocapsid protein, and so on and so forth. So what I'm going to do, I'm going to copy the sequence of the spike protein. I can very easily do it here using just Control C. And I am going to go to my internet browser. The way that I found this primer tree tool is I just Googled. If I go to Mr. Google, I can just ask it primer design tool, and you're going to be given several tools. In general, I use it IDT because my lab used it to buy oligos from IDT, but we can just Google primer 3, and that way you can find this link. Do you guys need that I put this link on the chat? It's better if I do, because I want to do it. We should do this together. Go ahead and open that link, please. Do you guys need that I paste on the chat the FASTA sequence of the spike protein?
Yes, please.
And to go back to SnapGene Viewer, I just copied, Control C. Let me first, so do you see the FASTA sequence? Do you see that I pasted the FASTA sequence?
Yeah.
Yes, we can see it.
And I do
Can
not
you
know,
paste it in the chat as well?
I don't know why it's not allowing me to paste.
Oh, okay, it's fine.
I don't know why it's not allowing me to paste. Let me,
Possible for you to share the file with us through
I
in the
shared.
chat box?
I shared the file with you already in the email that I sent yesterday.
Oh, okay, that's the thing. Okay.
Okay, what I can do, I can try to paste it on BME01. I have this open here. Do you see that? This file is also on the cloud already. It's the BME01. Do you see that? It's inside my folder.
Wait, Gio, I have a question. You said that there's something at the end of the email. It's like says GCF and then it ends with the genomic.dna.
Yeah, that's the
What
FASTA
is it
file.
exactly?
Fabrício,
Oh,
boa
okay.
tarde. Eu vou espelhar aqui, então, eu vou entrar, já entrei no Gal, tá? É no Gal mesmo que você faz essa
You can,
tirada?
Kira, you can download
Tá, eu vou compartilhar aqui, então,
that
tela.
sequence and open it using SnapGene Viewer, and then you'll be able to, you will not be able to get the
Deixa eu explicar o que está acontecendo. É que
though.
eu segui a
Because you
instrução
need
da
some
Rede
other
Vírus,
information for the spike protein.
fiz o download da planilha do mês lá no Gal, na parte de
need to mute.
Wait,
resultados.
so should I just go to the
Quando eu
BME01 doc
exporto aqui a
first?
planilha
Yes.
para mim, ela
For now, yes.
vem de acordo com aquele modelo que a Rede Vírus
I
pede.
just pasted the link in the chat for the
Então, deixa eu colocar aqui para você ver. Ó.
one in document.
Oh,
Tô
thank
aqui no
you.
Gal já. O que o professor lá me falou que tinha que vir aqui
I
nessa
have
aba
to
de
mute Jaime,
consultas, né?
but I don't know
Consultar
how to-
exame. Estou fazendo correto? Aí, por exemplo, eu vou botar aqui o mês de junho, só para a gente ter um exemplo. De 1º de junho a 30 de junho, porque ele cobra no máximo 30 dias, né? Vou filtrar aqui, ó.
Can you mute yourself?
Não, aqui tá errado, que eu botei julho.
Jaime, can you mute yourself?
Muito bem, ele abriu aqui.
It's very simple. I am sorry. Do you guys still see my screen?
No, we can't see it.
Okay, I just muted Jaime. All right, so we are back here. You guys now probably have the spike protein sequence, correct?
Yeah.
All right, so you take that sequence and I want to design primers to amplify that region. So I want to go to my internet browser and I want to open the tab that I was working on, which is the primer 3 tab. I pasted the FASTA sequence here. And what do I want to do now? What do you think we need to do now? Do we need to decide the size of our primers? Or do we need to decide the size of our amplicon? You're welcome to talk.
The size of the amplicon first, I
Yeah.
believe.
Yeah, the size of the amplicon. And let's find where it should be here in this page. Sequence ID, targets, excluded regions, product size ranges. What do you think? What do you think is the product size range? Is that what we want?
That would be the amplicon size,
Yeah.
right?
Yeah, that's what we want. Well, I don't want an amplicon that is 1000 base pairs long. For me, it's too long. Let's do something between 200 and 400. I'm just going to say 200. Let's actually do 200 to 300. So I gave it my spike protein sequence, and I told it that I want a 200s to 300s base pair long primer. So I'm going to pick my primers. So it gave us this thing, primer 3 output. It has information about the oligo. It tells me the start, the length, the TM, the GC content. I don't know what this is. I don't know what this is. And it tells me about the sequence. So what is start?
Wait, Gio, how did you get here?
How did I get here?
Like to this website.
Oh, you select pick primers. The
Yes.
button.
Oh, okay.
Pick
Perfect.
primers. I am picking the primers, correct? It's giving me the, okay, it's giving me the sequence. What is this? What is the start?
The number of the base pair that it starts at.
Yes, it is the number of the base pair that the primer is binding to, that the primer is hybridizing. Does that make sense? What is the length?
The number of base pairs in the primer.
Yes, in this case, 20 is the number of base pairs in the left primer. I call the left primer the forward primer, and I call the right primer the reverse primer. Then it gives me the TM, the melting temperature, the GC content, and the sequence. So what I want you to do now is I'm going to copy this. I'm going to copy the primer sequence. I'm going to paste it on SnapGene Viewer. So I'm going to go back to SnapGene Viewer as soon as I find it. And I'm going to go to primers. Do you see that? Do you see primers?
Yes, add primer.
Add primer. I paste the sequence. It's already telling me where it's hybridizing. I'm going to call this SIP demo 1 forward. It calls it the left primer. I am calling it SIP demo SIP 2020 2021 SIP demo 2021 forward. Add. It highlights to me where the primer is. I can go to that region. I can go to that region sequence. So it's here. Actually, I had done the same test yesterday. That's why there's another primer here. And yesterday, this was my second trial for whatever reason. Okay, SIP 2021, 1 forward, okay? Any questions so far? All right, let's go back to Safari, to my internet browser. The right primer now, I also call it reverse primer. Let's paste it. Primers, add primer. I'm going to call it SIP 2021. Reverse, because this is the right one. Oh, did you see that it aligned the other way around?
What do you mean by the other way around?
I'll show you. The first one aligned from left to right. The second one is aligning from right to left. Does that make sense? Or does that not make sense?
Yes, that makes sense.
Perfect. Okay, so what I'm going to do now here on SnapGene Viewer is I want to see the size of this thing, the size of my amplicon. So I am going to do this. It is telling me that this amplicon has 268 base pairs. Does that make sense?
Yes, because we set it to be from 200 to 300.
Right, exactly. But this number has to be exactly the same that is in the website. Do you agree? Because if it is not, there is something wrong, of course. 268. This is telling me the sequence size. That is the size of the region. And that's the size of the DNA region of the spike protein. That's also the included region. The product size, it is telling me, is 268. Okay? Is exactly the one that is that SnapGene Viewer tells us. Okay? Any questions so far?
What does it mean by product size? Is that like the amount of, like what the primer found? The amount of complementaries?
Okay, let me directly address this question by showing this slide. No, sorry. It's not going to be here.
Excuse me, Joe, did you call me? I had a problem of connection here.
No, I was just asking you to mute you because we were hearing your call, but I figured out how to mute you. To mute your mic.
All right.
It's all right.
I'm sorry.
Well, it's all right. No need to be sorry. Okay. Thank you, Jaime. I need to show you the basics of PCR, but let me show you this. Let me address Lucia's question. When I say amplicon size, I am talking about I don't think this is a good example. I am talking about the length of the bases that are going to be detected in a gel electrophoresis. So what we do in the gel electrophoresis is that we have a lane with markers which are molecules of DNA with known sizes in terms of their length. I was trying to see if in my picture I would say I would show the length of the marker here. As far as I remember, this is about 500 base pairs. This one lane here. This one lane is around 200 base pairs, which means that this amplicon, this is what the amplicon is. This amplicon is between 200 and 500 base pairs, because in the gel electrophoresis, I don't know if you guys remember, but we applied an electric field that caused the DNA to migrate, and then we were able to detect our amplicon. Does this make any sense at all?
Yes, it makes sense. Thank you.
Yeah, so how do I expect this thing to have 500 base pairs or whatever size it had here? How do I expect from my PCR design? And as I showed you, I compare that with molecular markers. That's also called ladders. That's also called ladders. Any other questions? So I showed you what a PCR result looks like. If you guys allow me, I am not going to, let me see. I'm just very quickly going. Are you guys all right with the primer design demo? If you're all right, I am going to very quickly revise these slides on PCR. PCR stands for polymerase chain reaction. So we design primers to be amplified by PCR. PCR amplification is done in cycles. So in each cycle, we have, I'm going to show you denaturation, hybridization, and extension of DNA that allows, for instance, here, 2 copies are being multiplied to generate what? 4 copies here, but more copies in cycle 3. Actually, this is going from, sorry, this is going from 2 to 8, I believe. That's what the, yeah, that's what the legend says. So what doing, these are the PCR reagents. You do not need to know that much about them right now. Yeah, you need to know that PCR steps are 3: denaturing, annealing that we discussed, and extension. So denaturing is the opening of the DNA double strand. Annealing is actually the hybridization of the primers. I guess we discussed this because why do the primers need to anneal? Because DNA polymerase is going to sit on them and extend the template. So the primers, they have this name because they are used as primers to the DNA polymerase. So the extension is just the addition of nucleotides and the finalization or the formation of the initial strand, or the copying of the initial strand. We use an instrument, a machine called thermocycler. And you see that in the screen of a thermocycler, we can set the temperature conditions of each cycle, and this is going to be used to amplify our DNA. So how do we analyze the product or the amplicons? We need to include the we need to run the amplicons on agarose gel. And this is what that looks like. So you have a power supply that allows you to subject your DNA sample to an electric field in an electrophoresis, in a gel electrophoresis. And that allows the DNA to migrate in your gel. I just showed you the design, basically the primer design. And these are results that I showed you because I wanted to address Lucy's question directly. There are different types of PCR. One is called touchdown. I am not going to explain about this. Quantitative PCR is important. Quantitative PCR means that you can follow the quantification of the DNA, which means that in each cycle, you have double the amount of DNA that you had in the previous cycle. But when you look at the final results like this, this is a PCR result after, I don't know, I don't remember exactly, but probably 30 or 35, probably 25 or 30 cycles here. So what happens to the intensity of the bands when I am at cycle, I don't know, 15, for instance? What do you guys think the intensity would look like at cycle 15? The intensity at cycle 15 is going to be lower or higher? Does anyone have any guess? The intensity is going to be lower, because at cycle 15, here are the cycles in the x-axis. At cycle 15, we have a certain amount of DNA, and quantitative PCR, we can couple the appearance, I would say, of DNA, the doubling of DNA, to a fluorescence level. Well, for your information, that's done through using a dye. So the simpler way that I explain this is that as soon as DNA is being born in the tube, it is increasing its concentration. So the dye stains DNA, and it's detected in the y-axis here. So as the cycles progress, the intensity is increasing because the amount of DNA is increasing. Does this make any sense at all? It doesn't make sense?
Yeah, I think it makes sense.
Okay, yeah, so that's the idea of what a CT value is. That's a cycle threshold where you start to be able to detect the DNA level. Okay, I am done with this. And now I need to show you the final part, which is the in silico PCR detection using the UCSC genome browser. Let's do one thing. Let's go to Google. Please do this with me. I will wait you guys to open Google. Type UCSC genome browser. Click that one option. This is what the UCSC genome browser looks like. On the tools, there are many tools that you could use. I'm not going to talk about any of them except for the in silico PCR. Does this make sense? We were not able to do an actual PCR, but let's do an in silico PCR. And here in the in silico PCR, we can do an in silico PCR using several genomes. You can use SARS-CoV-2, zebrafish, turkey, sheep, rhesus, macaque, rat, oh, sorry, shouldn't have clicked on it, pig, mouse, we can do mouse, chicken, elephant, oh, it's a shark actually, ferret, gorilla, eagle, human, where is human? It's not here because it's very special. It's at the beginning, it's the first. Yeah, human and mouse are very special. That's why they're at the beginning. Which genome do you guys think we should use today?
Let's do panda.
Why?
I don't know, they're cute animals.
They're very cute, but we do not have primers for them.
True.
do
Let's
we?
do COVID then.
Let's do COVID because, okay, one day we will do the panda to please you, Lucia.
Thank you.
Okay. And then they ask about an assembly. What is an assembly? Did I talk to you about assembly today?
Is that the de novo genome assembly?
Yes, great. The de novo genome assembly. Exactly. So that sequence that was assembled, well, in the case of the coronavirus, I don't think there was that need, because there could be. Actually, I am taking back, there could be, because the size is 30K. And I told you that Sanger, the average is probably around 1,000. So you cannot do Sanger doing 30K all at the same time. So there needs to be assembly. You probably cannot do that using Illumina, but I am thinking that it's not a problem to do that using Nanopore, because Nanopore average length is on the thousand. It's probably 20,000, 30,000. Should not be a big deal for Nanopore. Okay, yeah, perfect. So the assembly is that information. This assembly is the FASTA sequence. So the one that I sent to your email today is this one. Now what I'm going to do is I'm going to go back. Either I go back to SnapGene Viewer or I go back to this online tool here, and I copy my forward primer. I go back to the UCSC Genome Browser. I paste it here. And I copy my reverse primer, and I paste it here. I think this is the reason why I only call it forward and reverse, because that's what the UCSC Genome Browser calls it. Forward primer, reverse primer. Because why do you think I had to do so many in silico PCRs?
Well, you said that you were applying it to look at blood samples, I think. Or you were
Yes.
hoping to do so.
Yeah. Yeah, but I'm going to tell you that the reason why I did this so many times is because I wanted to make sure that the primers that I was using amplified the region that I expected. For COVID, that's very, very simple because it's only a 30K genome. But for human, the primers can be very unspecific, and sometimes they can bind to multiple sites, multiple places. So sometimes I need to be more stringent about the primer design, and I need to use a different tool. I need to think more about the way that I'm designing. But did you guys paste your results here? Do you want to do it? It's I want you guys to do it, please.
Your forward primer and your reverse primer are the same.
They are?
Yeah, I
Thanks, Sam.
I don't know if you made a mistake.
I have a quick question. Can you explain again the relationship between the forward and reverse primers?
Yes, that concept is related to the fact that we have double-stranded DNA. To tell you up front, the thing is that the forward primer is binding on one strand and the reverse primer is binding on the opposite strand. If they bound on the same strand, the PCR would never happen. Does that make any sense?
Yes, perfect.
Yeah. So I'm going to, do you guys, are you at this stage? Do you have your forward primer in that tab and your reverse primer in that tab? Okay, I'm going to submit. So it generated to us this here. It's it's telling us the assembly that it used. It is telling us the positions in the genome that the primers amplify. It's 24,000. From 24,000 all the way to 25,000. If you calculate, you'll see that it has 268 base pairs. It gives us the sequence of the forward and the reverse. And this is the amplicon. So when I showed you the band, like, I need to go back to my PCR thing. Let me ask you the way that I think about this. Do you see, if this was a band that amplified COVID material, do you see the letters here? It's a weird question because
No.
no, but they should be there. If this band corresponds, if that band corresponds to this band must have those letters. Does it make any sense, or it's just weird?
So as in they would show up to be the same, so you would know that they were the same thing.
What I'm trying to say is that this signal must come from the sequence that you are designing. This signal must come from the sequence that you are designing. If it does not, there is any problem, some problem.
So 1, 2, 3, and 4 all have a problem.
No,
then?
do not think about 1, 2, 3, and 4. 1, 2, 3, and 4, they have different sizes. They are not the same as this. This 5 is bigger than 3. 5 is bigger than 4. 5 is bigger than 2. And their sequences could be very different. Okay. I guess I am done with PCR. Do you guys have any questions, any comments, anything about anything that we did? Because otherwise I will move to, okay, I don't think I will go to the coding today. I'll have to start the coding tomorrow, or I can do it very quickly. What I want to do now, if that's okay with you, any questions, any comments, anything? Okay, I'm gonna close this because I'm gonna be done. I'm gonna call it a day in regards to this. What I want to do now, yeah, I'll save. What I'll do now is I will move to the coding aspects of each of your projects. Okay, the first thing that I want to say is that there is a better description now of each of your projects. And for BME1, I am the primary mentor. BME4, I am the secondary mentor, so I'm going to assist completely on the coding aspect. But I believe Jaime and Danilo will help a lot on the virology and epidemiology aspect. Same thing with BME5. I'll be the secondary mentor, but I'll mainly deal with the coding aspect, and Danilo and Jaime will deal with the virology and epidemiology aspects. So I'm going to start with BME 1 because hopefully people from BME 1 will be able to help the others, because I can try to give them as much of my expertise as possible so that they could help other people, because everything that we do here is going to be used on the coding aspect of BME 4 and 5. And I'm going to give you how I see that. You guys are going to ask your questions, comments, and I would like you to write on the session that I put after each. I'm going to remove the FASTA sequence from here. Is that okay? Because this was not intended for that. This session is intended for people to ask questions, and then I will respond. And if there is any better solution for this, I am very open to use some other tool for this. But the reason why I like keeping it as a Word document like this that everyone can edit is that we can keep track of it, in the sense that we can read it again and again and again until things make sense. Let's, at this moment, keep this only for the high level. We are not using this for questions on coding, for instance, but Gipoliano, Gio, am going to use this to read your doubts and your questions, and I might respond to your questions here or somewhere else, but I think this is the best place to start. Later we can move to Slack, but I think Slack is too much loose of a thing. It's too loose for us here who need some close and specific attention. So if you have any questions that you can type here that might help other people, I really encourage you to put it here. I do not care who you are asking the question, so you do not need to put your name, and I'm actually not sure if this is going to track who asks what, but I'm not interested in tracking who asked what, and to be perfectly frank, I cannot keep track of 11 students asking me these things. So my intention is that a question that you ask here might help someone that might have the same question later, okay? So you can read everything that I already wrote specifically to BME 1, BME 4, and BME 5. But the very high level for BME 1, we already did primer design. We are going to do SNP identification using FASTA sequences, and that's going to be covered partly. I believe it's going to be on the session next Thursday. The three of the groups are going to take advantage on that, because BME 4 can use the command grep, for instance, to identify mutations present in the VCF files that are produced in the bash pipeline. So I need to tell right now Angelina, Jeremy, Lucia, and Lynn to help me with bash, because we will use bash to produce the VCF files. Is that relatively clear?
Yes, it's clear.
So from I should have written it here somewhere, or it should be somewhere to you. that from the VCF files, we can basically grab any mutation that we want. And to tell that up front, we are going to grab mutations using the nucleotide coordinate. And then one thing that is possible to be done with that is let me see. Okay, so you see from the pictures here, the last year, people constructed these databases, but really this database here is just an Excel sheet, an Excel spreadsheet, right? Where you have information about the continent, the country, the sequence, the size of the size of the file. In this case here, we were using FASTQ files. We were not using FASTA files. This year, the only way that I can work with you is by using FASTA files only. There's no chance that I can help you using FASTQ files, but I need you to know how, I need you to have an idea how this is done. And you can see here that we use it. So let me tell you, we use it the sequence read archive, the SRA files, and we store the information about them in this database here. So basically, the way that I can explain is that we are building a database to store information. And one thing that we can do here, one thing that is a deliverable for you guys, for Angelina, Jeremy, Lucia, and Lynn, is a visualization of the VCF files using a genome browser. I do not care if you use IGV. I do not care if you use UCSC genome browser. You can visualize a VCF file using a genome browser. And this is one visualization tool for that that you can use for the VCF files. Questions, comments, doubts? We're going to go through the command line steps of generating VCF files, okay? All right, I think I will move to BME 4 now. So BME 4 is bioinformatics.
I think that Kira has a question, sorry.
Oh, go ahead, Kira.
Oh, I just had a question about how the difference between the FASTQ and the FASTA files and why we would be using FASTA files instead for this year.
Okay, the FASTQ files are used to assemble a FASTA file. So in this perspective here, FASTQ files do not have any usage for us. We could use FASTQ files to do variant call. It will just increase the amount of work for me, which is something I don't want. Does that make sense?
Yeah, okay.
Well, yeah.
All
Yeah,
right, thank you.
Sure. Yeah, the pipeline, I can give you the pipeline for FASTQ variant call. It will just be harder on you and on me. Okay, any other, but on the other hand, Kira, if you want to work on the other thing that I forgot to say, and I should say, is that I can give you guys access to these Excel spreadsheets. You can improve / increase / refine this database construction. And if you guys want to use that for publishing in Columbia Journal, high school journal, or undergraduate science journal, you're more than welcome to do that. Okay. All right, let
Sounds
me
great. Thank you.
let me move on to BME4. So the title is Bioinformatics Pipeline to calculate the frequency of viral variants of concern versus time in a geographic location. So on the previous project, what people did was they identified variants in regions. They identified variants in Brazil, in China, in I think Australia, some countries in Europe, in Africa, and in here we are going to calculate the frequency of mutation in each region. So at the very high level, what you guys do here is you take what I think the way that I will do it next week, we are going to take a look at 250 sequences from Brazil. How many of those sequences have the mutation at position 23,000? That's what you guys are doing here. So in Brazil, we have, I don't know, 100 of these 250 mutations, 100 of these 250 samples with mutation in this position. That's, I don't know, 75% or 30% of what the samples represent. So with that calculation, we will know what's the frequency of each mutation in each region. And that has to do with the concept that Professor Danilo discussed with you of the incidence of a mutation in a certain region. Okay, I think you guys need to read this. So you may take a look at I can give you access. You guys need to talk among yourselves and tell me if you want access to this, if you want access to the work that the group last year did, and if it's going to be useful to you, because I just do not have the time to go and check that for you, basically. So this is the regional SNP distribution. If you look at I hope you realize that this is a slide from the presentation last year. It's basically an Excel spreadsheet of the mutations. These mutations are from a paper that had described the COVID mutations. It's called the IN paper. And if you do not ask me for this paper, I'll probably give you later during your process. I just need you to ask me for the IN paper, and I'll give it to you right away, or it should be in the references of people that worked last year. So what people did was they calculated the frequency of each mutation in each region. And that's basically, you can treat each of these mutations as the variant of concern for now. And then what people can do here also is they can do the hierarchical clustering that the students did last year. And the hierarchical clustering is through the plotting of the heat map using the data from last year. I am very happy to give you the code that people used for last year, but obviously you cannot use their work from last year. So you need, basically, I need you to work on your region of interest this year. And hopefully people will get to discuss a little bit on what differentiates a hierarchical clustering from phylogenetic analysis. How is this different from the phylogenetic analysis that Danilo discussed with you last week? So I wrote some specifics here for you guys. They also did the phylogenetic analysis last year. Okay, this does not belong in here. This is the application. And you guys are also very welcome to put your questions. I mean, BME4, very welcome to write your questions here so that Danilo, Jaime, and I can look at them and have a sense of what your questions are. And I hope we'll figure out a good way to address the coding questions. Again, I have not had the time yet to think about a platform where we can write coding questions. I am thinking that GitHub is a good way to do that, but I have just not figured out how to do that using GitHub. Now moving on to BME5, which is building a web application for visualization of SARS-CoV-2 variants of concern for use in the context of a middle-income country. Obviously, here I was thinking of Brazil because that's where I come from. And the reason for this project is because I was funded by public investment from Brazil. So I need to work on inventing things that could possibly help the country that I come from. So the basic for this project is that. So there's the description here, the tasks, and the reason why there are so many tasks here for you guys is because this is pretty new and an ongoing project. You are also welcome to write all the questions that you guys might have in the session here. But one thing that I want to show you is that first of all, I want you guys to read this online paper. So let me go here and put that on the browser so that you can see. This blog is called Towards Data Science. I have read material from this blog before. They have very good material about data science, obviously. And this article is how to build a data analysis app in R Shiny. So Shiny is an R package used for building web applications. They discuss things here and they provide code through GitHub that you can test for building your app. And what we had to do, what we had and have to do for BME05 is we need to understand their code and adapt their code for the needs that we have in this project. So first, you guys need to read that article. The link is here. We need to decide. We need to talk about these deliverables for you guys. Either you guys provide us with visualization of the data that you analyze for the app as it is. And I'm going to show you in this website here how the app is right now. Or you work on the development of the code for a visualization. And that's something that Esther in Brazil, I and Jaime are talking about. We are still developing this code. And this is a visualization. Actually, the part on the right is a visualization of how the software is plotting the analysis that we are looking at. Actually, these are, I think these are mutations that the software is plotting. So we need to make an agreement on what the deliverables are going to be for BME05, whether you guys are going to just look at the visualization that you do yourself, or whether you're going to decide to work on the R code. And we do not need to talk about that now. We need to talk about that probably for a week from now, more or less, which direction you want to go. So now what I'm going to do is I'm going to show you what the application looks like. The application is hosted in this website that I paid someone to develop for me, because that has to do with my public funding in Brazil. So I'm going to type that here on my web browser. So this, I call this initiative, I'm going to state it in Portuguese, because this is basically for the government of Brazil to see. So this is called DNA Brasil. And it's the idea here is that people would come and they would do some visualization using the tools that we develop. So the stage that we are at right now, this is written in Portuguese again, because the public here is intended to be Brazilian. So what this application is doing is calculating the frequency of mutations, the phylogenetic analysis, and what we are calling here line graph. And the line graph, the way that it is right now, my perception is that it is showing the increase in variants of concern. You can use it basically for visualization of variants of concern. So to do a very simple demo to you, you can download this SARS-CoV-2 FASTA sequence and try it. This file here, I think it's going to be corrupted as of right now, because there are problems that we are addressing, but you're very welcome to download this and try it. But I already downloaded, and I am going to browse the sequences that I downloaded. So if I try with that one sequence, I'm going to have the visualization, the table. We are working on the table of frequencies. This is the way that it is right now. The phylogenetic analysis is okay for the purpose of the demo. The line graph is not okay. Clearly, it's not interpreting correctly some information on the sequence that we input. And because we worked on that, what I'm going to do now is I'm going to browse this other file that is our, this is, I think they call this a toy example. This is another sequence file. You can see here the table, the frequency table again. It's working for the phylogenetic analysis, and I was under the impression that it was not working for the phylogenetic analysis, but it's working okay. It's also working for the visualization of the graph of lines, but clearly there are many things here that are not working the way that they should be working. So obviously we need to work on the code that is doing this. Yeah, so here are the credits for this. You guys are welcome to work either on the input to this or the code that makes this be available here. So I'm going to go back here. Again, on the right is a visualization of what the frequency of variants is looking like right now in using the software. On the left is my intent, is my goal. My goal is for it to be like this. Yes, I am done with this part. Do you guys have any questions about anything that I said? Is everything clear enough? Okay. Let's just get started on this, because I just feel that we must get started. You all should know where these recombio general instructions are, correct? Do you know where this link is? Now I can see.
They
your
were
face.
sent in an email, in a zip email, so yeah.
Okay, I need you all to see these slides. There are instructions here. This is what our Markdown looks like. I'm going to revise this on Tuesday, but I need you to be aware of this starting right now, because we're going to insert each chunk where we can write R and bash code. Okay, so you can download the code that we are going to use. This slide talks about downloading the code that we will use from GitHub. Did anyone has anyone tried to download the R code?
Yes, I downloaded it and it's perfect.
Great.
Like it works perfectly.
Were you able, Lucia, to knit without any problems?
To what? Sorry?
To knit. Oh, did you try R Markdown or not?
That was a couple weeks ago, so I can't
Okay.
remember honestly. Sorry.
Okay, that's fine. We need to revise. This will come again tomorrow, Monday, and on Tuesday. I wrote this slide as a personal note. Do not do this, because I myself have messed my computer with GitHub because I didn't fully understand what it was doing, and I deleted things that I didn't want to delete. So do not use this. But later I hope I will mature enough to show you how we can take advantage of all of the resources that are here. Okay, what I need to know now, I need one of you. Has anyone, this is going to end. People that need to leave can leave right now. That's okay. I just need to see what is your guys' ability to have R or not, and particularly R Markdown. I see that Kira has a question. Go ahead, Kira.
Yeah, I think I was gonna ask Lucia where she had, because I couldn't find the slides right away.
I just
Lucia,
sent
is
it
that
to
the
the
link?
chat.
Oh,
Yeah, I
right,
just sent the
thanks.
link.
Yeah, so people that are on Slack, the Recombio Slack, they should have access to these slides. And I guess for me, it's going to be the easiest way, because let me tell you, I have the 10 of you. I'm going to have the 10 of you Tuesday, 10 a.m. I'm going to have apparently 60 people registered for English at 3 p.m. And I was not expecting that. And I think it was 8 people that registered for 3 p.m. with, I think it's 3 p.m. California time. And 5 p.m. is the Spanish. 8 people registered for that. So I am going to have a hard time, I think, from the standpoint of dealing with different people and different codes. So this slide should be on the Recombio Slack.
Oh, is there a link to the Recombio Slack? Because I'm not sure whether I got the link to join. Maybe I wasn't here.
I have it open here. Do you want me to share it?
Yes.
Sure.
Lucia, please, if anyone here in this cohort complains that they are not on Slack, I encourage you to add them to Slack. And if you do not have permissions, we have to ask Sierra to give you permission, or that I give you permission to add people. Because it's going to be hard for me to add one by one of 80 or however many people to that.
Yeah,
Okay.
so I just copied the link. I'll send it here and you can tell me if it works to join.
Yes. All right. Yeah, we are figuring these things out, and I am figuring out the best way to address all this. All right, so great. Kira, did you have another question?
Oh, I think I'm good. I was just gonna ask, is the R Markdown separate from R in general? Or I already have R and,
R is a language. R as a language you use if you download just R, the program. RStudio is, I don't know if it's a graphical user interface, but I think it's a better interface to deal with R. And really, R Markdown is an even better interface. And I think R Markdown is just a format, is actually a text format for you to deal with R. So what you can do in R Markdown is you can insert chunks and then write R code, insert another chunk and write Python code, insert another chunk and write bash/Linux code. And what I love about that is that as you do this, you put your notes and then your code is reproducible. You can know what you were thinking when you were writing in R or in Python or in Bash, and you get that idea, and then you get to repeat your results. You can write about the locations, you can write about the problems that you had, for instance, and that makes your flow of thoughts way better, in my opinion. You want to ask again, Kira?
yeah, I was going to ask about, so I have both R and RStudio downloaded. So is R Markdown separate from those two, or am I all set?
No, that's okay. That's one thing that I wanted to address, and because this is going to be recorded, I think it's going to be useful for people that come back to this. Would you mind sharing your screen with me so that I can? The thing is that you should be able to use R Markdown right now because it's just a format in RStudio. Do you want to share your screen with me? I think I need to give you permission. Do you want to share your screen with me, or you cannot?
Oh, how should I do this? Because I just have it under applications. As in, I actually downloaded it a long time ago, like before this project started.
Did you download R and RStudio?
Yeah.
Can you share your screen with me? I just want
Yeah,
to I just want to use your interface as a toy example.
Yeah. So I have
click on
RStudio.
RStudio. Click on RStudio, please. I need it to open.
Yeah, I know it's loading.
Okay. Is it not open?
It's opening. It's just being really slow. All right.
Perfect. Yeah, there's always it always can take some time. Okay, so did you, ask it to open a script or something?
Yeah, I just have it under applications. As in, I actually downloaded it a long time ago, like before this project started. Do you want me to share my screen?
Okay, so did you ask it to open a script or something?
No, I didn't. I think this is the first time I've opened it. I think I downloaded it and it didn't use it.
Okay, I will test two things in your computer. Can you do where in your screen the green plus sign?
Here?
You can click that. That's going to maximize.
Okay. The green plus your new file?
Yeah, can you go file, new file,
Yeah.
text
And-
file, text file. I always do it as text file, and I think it's going to, I'll have a problem. Okay, can you save it? Save that document. We are going to save it with the format R Markdown. Save as, you can call it title if you, test if you want.
Okay.
Dot R MD for R Markdown.
All right.
Save it wherever you want to save it.
Okay. I'll just save it in downloads.
Yes, perfect.
Okay.
Perfect. Do you see that the icon changed to that purple thing? It should say RMD. That means that now we are using what I call an R Markdown. So let's try one thing here. Can you hit enter? Can you click on insert, please? R. That's what an R chunk is. And what that does is that you can insert R code in there. Can you go, if you go after the 3 dots at the bottom? Yes, enter again. Insert. Bash. Oh, okay. Can you type there CD? I'm going to list. Can you type tilde, space, tilde, slash, desktop, capital D. Can you click on the arrow, the green arrow on the right?
Run.
Where it says bash, there, and the right part.
here.
Yes, yes.
Run current chunk. Oh. What does that mean?
It means that there is an error.
Yeah, but I don't think we ever referred to a package, did we?
No. Let me see. I need to get that from my code. See, these are the these are the problems that I'll start dealing with more and more. Can you type? Let's see. On R, in R, can you type install dot packages? Yeah. That one, knitr. See if it runs.
Wait, just click here.
Yes, yes. Can you put colon? No, not colons, quotations around knitr?
Single quotes or double quotes?
Double. Run again. You don't have a lot of memory. Your computer
Yeah.
doesn't have a lot of memory.
Yeah, I think that's also the reason why the other thing said I didn't have enough space left.
Yeah, but it's going to be a problem for others. So it's good that this is being recorded here so that others see it too. And yeah, thanks everyone for staying, because you are going to run into these problems. And this is, I am sorry to say, this is 80% of dealing with computational biology.
So is the bash the same as using terminal on the Apple or the I guess on
Yes,
the Mac OS? Yeah.
yes, yes. Yes, basically. But I prefer to do it from the R Markdown because I can document everything that I did.
Oh yeah, because once you close the terminal, it closes the session, right?
What do you mean?
Oh, I've used terminal before, and it's like once you close that window, then it'll just when you open a new window, it'll just be like start again, completely
Yeah.
blank.
Yes. Can you? It's not done.
Should I just stop the thing or?
No, I want to see how we can solve this and spend some time on troubleshooting, because everyone is going to complain about these things. Let me see. There should be, can you on the right, can you click on packages? Can you go down to see if you find neither?
I think that's it. There's only one under K, and it's the turn smooth.
Yeah.
Should I check for package updates?
Try.
I did.
Can you click on the button install?
And then
Packages. Try to see if you can do neither.
Oh, there it is. We just want plain knitter, right?
Yeah. Install. Nothing happened?
Yeah, I think I clicked it. Oh, what just happened?
Do you uninstall? Do you uninstall from sources the package which needs compilation? Yes, no, cancel install. Can you go? On the console now, where your cursor is blinking, can you type library? Knitr? Or you need
And
to
that?
open parentheses.
Oh. So type knitter on the inside.
Yeah.
Like that?
Yeah, hit enter. It's not installed.
I think maybe, was I supposed to say yes here? Like,
Where?
after do you want to install?
Did you say no?
No, I think we never replied.
Can you try installing it again? Hit install
Yeah.
again.
Okay, so this popped up.
Do you uninstall from sources which needs an application? I guess you need to say yes there because I don't think you have anything.
Mhm. Is it still going?
Yeah, it's down. Do you see the stop sign?
Yeah.
It means that you could click on there and stop this process.
Oh, there it is.
Great. Can you type library neater? Great. So what you just did was you uploaded the library to the R environment. So in the R environment, you have neater now, so you would be able to use it. So we did this because Bash complained about no package called neater. Can you now type C? Can you now run that Bash thing? Great. What it just did was it changed directory to the desktop. So what's going on inside of that Bash chunk is in there, you are inside your desktop. So can you go to the line where it says desktop? Do you mind if I list everything that is in your desktop?
Oh, that's fine.
Okay, just give an enter. You need to be inside of that chunk.
Okay, yeah.
Yes, you can type ls for list and star. Star means everything. So if you run that again, yes, okay. These are the things that are in your desktop now. And it looks like you have Bash. You would be able to run, for instance, the alignment, BWA alignment, using a chunk like that. So next week, when I show you guys how to run BWA using Bash, you can do that. And so because of that, other people that watch this recording would be able, I will know now that I told people how they can run Bash in their RStudio interface. Is that relatively clear? Do you have any comments, Kira?
Yeah, I was just also wondering how, like, is there a better way to also get out of the application once you're in here? Like, do I need to go back out of the desktop or?
What do you mean?
Or like for terminal, you just end up clicking out, right? Like you quit terminal and then you're done. I was just wondering if there's like anything you need to save before you leave or quit R or something.
No, as far as I know, you can just save that and your code is going to be there forever.
Oh, okay, great.
Yeah. Yeah, that's, I guess that's what I meant by you just save this and your markdown notebook is going to be saved wherever you want it. Can you go, did you save it in
Yeah.
your desktop?
No, I saved under downloads, I think.
Yeah, so if you CD to download, you should be able to list it there. Is that what you want to do?
Sure. Well, we should see it there, right?
Yeah,
Is downloads also capitalized?
yeah.
Okay.
But you need to ls everything.
Oh, yeah. Should I delete the top part or?
Yeah.
Okay. Oh, there's a lot of stuff here.
It's ordered in alphabetical order. It's sorted.
Oh, there it is.
Yeah.
Okay, great.
Perfect. 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 So we are going to
That's-
deal with
Oh, go ahead.
we are going to deal with these things more closely last week. And as I said, from now on, 80% of your time is going to be debugging these issues. All right. Do people have questions, comments?
Oh, I was just gonna say that looking at the number of downloads, they're just like random images. I think that I figured out where I can save more memory on my
Yeah.
computer.
Yeah. Okay. Let me show you this. If you go to your other line.
Oh, the R
No.
part?
No, the bash.
Oh, yeah.
Go down. Go to
Mhm.
another line.
Oh, another one. Okay.
In that chunk, type du space dash sh. Sorry, there's no space before the sh.
Oh, okay. Yeah.
And then space tilde backslash downloads. You're gonna see the size of your thing.
Okay. So just run it.
Run it, yeah.
It'll be at the bottom, right?
Yeah.
What?
Can you go up?
Wait, where would it be?
Good question.
Is it still calculating it?
No.
Maybe it's at the.
Can you remove cdls and leave it and run it again?
With this?
Yeah,
Oh.
there it is. Surprisingly, there is not much there.
Oh, huh.
If you do that for me, you'll see 20 gigs, 100 gigs.
Really?
Yes,
Wow.
that's why I need to have an external hard drive that I can store 1 terabyte of data on it. All right.
Okay.
The purpose of this, everyone, was for you to be familiarized with Bash, basically, and with R and with R Markdown. So I hope that others see more or less what we are going to be doing. And I guess we are all good to go now. Thank you so much, everyone, for joining. Thanks, Sini, for letting me know that you need to go. I'll hold a session tomorrow at 10 because I had told people that I would do so. But if I am asked, I will try to manage to have another session probably in the afternoon. And I hope that people can join the sessions on Monday so that people do not complain so much about computational biology or coding for next week and for the weeks after. All right. Thank you so much. I'll see you next week or tomorrow or whenever you want to come see me again. All right. Thank you.
Thank you.
Thank you, Dio. Bye.
Thank you. Bye-bye.
Bye-bye.
Bye-bye. Peraí que eu vou parar de... Let me stop.